Welcome back to Rad Onc Smart Review, RadBio edition.
Great to be here.
So today, we're really diving deep into tumor suppressor genes. These are kind of the cell's natural defense system, right? The brakes against cancer.
Exactly. They're the vital components keeping cell growth in check. Think of them as the cellular brakes.
Okay. And we're going to explore what happens when they, fail.
Yeah, precisely. We'll look at what happens when they falter, get into Knudson's two-hit hypothesis, which is, well, pretty foundational stuff.
Right.
And we'll talk about some key players, genes like RB1, TP53, BRCA1 and 2, PTN, APC.
The big ones?
The big ones, yeah. And importantly, how all this connects back to what we do in radiation oncology, some real clinical relevance.
Perfect. So let's start at the beginning. What are these tumor suppressor genes actually doing inside a normal, healthy cell?
Well, fundamentally, they're normal genes. Their job is to make proteins that regulate how cells grow and divide. They prevent that uncontrolled proliferation that, leads to cancer.
So they really are like the brakes, or maybe guardians.
Both are great analogies, actually. They act as brakes on the cell cycle, but also as guardians, constantly monitoring the cell's health.
Okay, and how do they do that? What are their main jobs?
They have a few critical functions. First, they can slow down or even stop cell division. They act as specific points, checkpoints in the cell cycle.
Like quality control points?
Sort of, yeah. Making sure everything's okay before the cell duplicates its DNA or divides. Second, many are involved in DNA repair, fixing mistakes or damage to our genome.
Which happens all the time, right?
Constantly. So they maintain genomic integrity, keep things stable. And third, if a cell is just too damaged or starts acting weirdly.
Beyond repair.
Exactly. They can trigger apoptosis.
Program cell death, the self-destruct sequence.
Precisely. A crucial way to get rid of potentially dangerous cells.
Okay, so this is different from oncogenes, which we talked about before. Oncogenes drive cancer when they become overactive.
Right, like a stuck accelerator.
Whereas tumor suppressor genes cause problems when they lose their function, when the brakes fail.
Exactly. It's a loss of function mechanism. And when those safeguards fail, cells can escape the normal controls, multiply unchecked, and, well, eventually form a tumor.
Right. And that loss of function brings us to Knudson's idea, the two-hit hypothesis.
Yes,
Yeah.
exactly. A really foundational concept. It came from Dr. Alfred Knudson back in 1971. He was studying retinoblastoma, a childhood eye cancer.
What did he notice about it?
Well, he saw two patterns. Some kids had it hereditarily, multiple tumors, often in both eyes, showing up really early. Others got it sporadically later in life, usually just one tumor in one eye.
Interesting difference.
And Knudson proposed that to really knock out a tumor suppressor gene's function, you need two separate events, or hits. These hits have to affect both copies, both alleles of that gene in the same cell.
Okay, that makes sense. We inherit two copies of most genes, right? One from each parent.
Exactly. So usually having just one working copy of a tumor suppressor gene is actually enough to do the job to control growth normally.
Ah, so losing function is recessive, at the cell level, I mean.
Precisely. At the level of the individual cell, you need to inactivate both copies for the cell to lose that control and start down the path towards cancer.
And this two-hit idea explains those different patterns he saw, the hereditary versus the sporadic cases.
It does, yeah. It explains them beautifully. So in sporadic cancers,
The ones that just pop up randomly.
right, both hits are somatic mutations, meaning they happen after conception during a person's life, just by chance in a particular cell. Maybe one mutation hits one allele,
Okay.
and then later, maybe years later, a second independent mutation hits the other allele in that same cell, or one of its descendants.
So it takes two unlucky events in the same cell line.
Exactly. That's why sporadic cancers linked to these genes tend to show up later in life, and usually as a single tumor.
Okay, makes sense. How does that contrast with the hereditary cancer syndromes then?
In hereditary syndromes, the person inherits the first hit. They get a mutated non-functional allele from one parent through the germline, so it's in every single cell of their body from birth.
Wow, okay. So they start life with one strike against them in every cell.
You got it. Now, they only need one more event, a single somatic second hit, to happen in any susceptible cell to completely lose that gene's function in that specific cell.
And because that first hit is already there everywhere, the chance of a second hit happening somewhere is much higher.
Much, much higher. Which is why these individuals have a significantly increased lifetime risk of developing certain cancers.
And they often get them earlier.
Yes, often much earlier than sporadic cases. And you also see multiple independent primary tumors or bilateral tumors, like in both eyes for retinoblastoma, or both breasts, or both kidneys, depending on the gene.
So let me make sure I understand this. In the cell, the mutation is recessive, you need 2 hits. But the syndrome, the predisposition to cancer in the family, that looks dominant when you track it.
That's a really key distinction, yes. You've nailed it. The loss of function in the cell requires both alleles to be hit, making it recessive at the cellular level. But the cancer predisposition is inherited in an autosomal dominant pattern.
Why is that? Just because the second hit is so likely?
Exactly. The probability of acquiring that second somatic hit somewhere, sometime during life, is so high for someone who inherited the first hit that the cancer appears to follow a dominant inheritance pattern in families. It seems like just inheriting one bad copy leads to the disease, even though mechanistically 2 hits are needed in the cell that becomes cancerous.
Okay, crystal clear. Now, how does that second hit typically happen? Especially in someone who inherited the first bad copy.
Well, a very common mechanism is something called loss of heterozygosity. We often just say LOH.
LOH. Right. Heterozygosity means having 2 different versions, 2 different alleles, right? Like one normal and one mutated.
Precisely. So loss of heterozygosity is when that cell, which started out heterozygous, somehow loses the remaining good, functional, wild-type allele.
So it ends up with only the bad copy, or maybe 2 bad copies.
Exactly. The cell effectively becomes homozygous for the mutant allele, or sometimes homozygous if, say, the whole chromosome chunk with the good allele is lost. Either way, the normal function is gone.
And how can that LOH actually happen, genetically speaking?
There are several ways, actually. One is mitotic recombination. During cell division, chromosomes can swap bits, and sometimes a cell can end up inheriting 2 copies of the chromosome segment carrying the mutation.
Okay.
Or you could have chromosome loss. The entire chromosome carrying the normal allele just gets lost during division.
Triple deletion.
Or a smaller deletion, yeah, just taking out the segment with the normal gene. Another way is just a new point mutation happening in that remaining normal allele, inactivating it directly.
So a second independent hit, basically.
Yep. And finally, there's epigenetic silencing. This is where the gene itself isn't mutated, but it gets switched off often by adding methyl groups to its promoter region, a process called hypermethylation. It's like a molecular off switch is flipped on the good copy.
So LOH is really that critical second step, the event that pushes a cell over the edge in many hereditary cancers.
It often is, yes. It's the somatic event that finally eliminates that last bit of protection for the tumor suppressor gene in that susceptible cell.
Okay, this is fascinating. Maybe we should talk about some specific examples. You mentioned RB1 earlier.
Yes, RB1, the retinoblastoma 1 gene, foundational stuff.
What does its protein, the RB protein, normally do?
RB is a master regulator, a real gatekeeper for the cell cycle. Specifically, it controls the transition from the G1 phase into the S phase.
S phase is where DNA gets copied.
Right. RB does this by grabbing onto transcription factors called E2F. Think of E2F as the key that turns on the genes needed for S phase.
Okay.
When RB is holding on to E2F, E2F can't turn those genes on, so the cell stays in G1. But when the cell gets the right signals to divide, RB gets phosphorylated. Think of it like getting a tag.
Like a release signal.
Exactly. Phosphorylation makes RB let go of E2F. E2F is then free to switch on the S-phase genes, and the cell cycle progresses.
So if RB1 is mutated and doesn't work,
then E2F is basically free all the time, constantly telling the cell to enter S-phase and divide, even when it shouldn't. That's uncontrolled proliferation.
And that leads to familial retinoblastoma if you inherit a faulty RB1 copy.
Correct. That's the classic example. But RB1 mutations are also found very often in sporadic cancers too, things like osteosarcomas, small cell lung cancer, bladder cancer. It's really important across the board.
Okay. How about TP53? You hear about P53 all the time. The guardian of the genome.
Ah, yes. A very fitting title. The P53 protein is a transcription factor, and it gets activated by cellular stress, especially DNA damage.
So when things go wrong, P53 steps in.
Exactly. Once it's activated, it can do several protective things. It can halt the cell cycle, usually at the G1 checkpoint, by turning on another gene called P21. This gives the cell time to repair the DNA damage.
Nice time for repairs.
Right. If the damage is too bad, P53 can actually trigger the DNA repair machinery itself, or push the cell into senescence like permanent retirement. It stops dividing forever.
Or the ultimate failsafe.
Or, if the damage is truly irreparable, P53 triggers apoptosis. It activates genes like BX, PUMA, and OXA that basically tell the cell, okay, you're too damaged, time to self-destruct for the good of the whole organism.
Wow, so losing P53 function must be really bad.
It's incredibly detrimental. The hereditary syndrome is lifelong mini syndrome from inheriting a TP53 mutation. It causes a massively increased risk for a whole spectrum of cancers: sarcomas, breast cancer, brain tumors, leukemias, adrenocortical cancers, often hitting people very young.
That sounds devastating.
It is. And importantly, in sporadic cancers, just looking across all human cancers, TP53 is the single most frequently mutated gene. Over half of all tumors have a TP53 mutation.
Over 50%. That's huge. Really highlights its role.
Absolutely critical.
Okay, let's move to BRCA1 and BRCA2. People know these in relation to breast cancer risk?
Yes, the breast cancer susceptibility genes, BRCA1 and BRCA2 proteins, are crucial for DNA repair. Specifically, they work in the homologous recombination pathway.
And that pathway fixes what kind of damage?
Double-strand breaks. These are really dangerous breaks across both strands of the DNA helix. Homologous recombination is a high-fidelity way to repair them accurately, using the sister chromatid as a template.
So BRCA proteins are like specialized repair crew for major DNA damage.
That's a good way to put it. They're also involved in some other things, like controlling transcription and cell cycle checkpoints, but that DNA repair role is key.
And inheriting a mutation leads to
Hereditary breast and ovarian cancer syndrome, or HBOC. Germline mutations in either BRCA1 or BRCA2 dramatically increase the lifetime risk for breast cancer in women and men, and also ovarian cancer, plus increased risk for prostate cancer, pancreatic cancer, and melanoma.
So understanding these is vital for counseling and prevention strategy.
Absolutely. Prophylactic surgeries, enhanced screening, it all stems from understanding this risk.
Okay, next on the list was PTEN. What's its job?
PTN stands for phosphatase and tensin homolog. It's an enzyme, a phosphatase. Its main job is to put the brakes on a very important signaling pathway called the PI3K ACT pathway.
And that pathway does.
It promotes cell growth, survival, proliferation. It's a major pro-growth pathway. PTN acts as a negative regulator by removing a phosphate group from a signaling molecule called PIP3. By doing that, it dampens down the PI3K ACT signaling.
So it counters pro-growth signals. Another break.
Exactly. And when you lose PTN function, that PI3K ACT pathway can become hyperactive, driving inappropriate cell growth and survival.
What conditions are linked to PTN loss?
The main hereditary syndrome is Cowden syndrome, sometimes called multiple hamartoma syndrome. People develop lots of benign growths, hamartomas, but also have increased risk for certain cancers like breast, thyroid, and endometrial cancer.
And in sporadic cancers?
PTN is lost or mutated very frequently in many types, including glioblastoma, a type of brain tumor, endometrial cancer, and prostate cancer. Again, highlighting how critical controlling that growth pathway is.
Okay, one more major example, APC.
Right, APC, Aganomonas polyposis coli. Its protein product is a key negative regulator of another signaling pathway, the Wnt pathway.
Wnt signaling, also involved in growth.
Yes, very much involved in cell proliferation and differentiation, especially in the gut lining. The APC protein is part of a complex, often called the destruction complex, that targets another protein called beta-catenin for degradation.
Beta-catenin, what does it do?
When beta-catenin isn't degraded, it goes into the nucleus and turns on genes that drive cell proliferation. So APC's job is basically to keep beta-catenin levels low, preventing that signal from being constantly on.
So if APC is mutated?
The destruction complex doesn't work properly. Beta-catenin builds up, goes to the nucleus, and drives uncontrolled proliferation, particularly in the colon lining.
And that leads to the hereditary syndrome.
Familial adenomatous polyposis, or FAP. People with FAP develop hundreds, sometimes thousands, of precancerous polyps in their colon. And without intervention, like removing the colon, progression to colorectal cancer is almost inevitable. It really is. And we've only covered a few key examples. There are many others: WT1 and Wilms tumor, NF1 and NF2 and neurofibromatosis, VHL and von Hippel-Lindau disease.
And the mismatch repair genes in Lynch syndrome.
Exactly. MSH2, MLH1, and others involved in Lynch syndrome, or HNPCC. Each one plays a unique, vital role in guarding the genome or controlling cell growth.
So understanding all this fundamental biology, how does it directly impact us in the clinic, especially in radiation oncology?
It has huge implications. First off, risk assessment. Identifying people with germline mutations is critical.
Right, for genetic counseling.
Exactly. It lets us inform patients and families about their risks, guide decisions about increased surveillance like more frequent colonoscopies for Lynch syndrome or mammograms for HBOC, and even discuss prophylactic measures.
Like the preventative surgeries for BRCA carriers?
Precisely. Prophylactic mastectomy or oophorectomy can drastically reduce cancer risk in those high-risk individuals.
And what about using this information for diagnosis or prognosis once cancer develops?
Well, finding specific somatic mutations in tumor suppressor genes within a tumor can sometimes help confirm a diagnosis or classify a subtype of cancer. And definitely, they can be prognostic.
Meaning they predict how the cancer might behave.
Yeah. For example, as we mentioned, certain TD53 mutations are often linked to more aggressive disease and poorer outcomes in several cancer types. It gives us clues about the tumor's biology.
And are we getting better at actually treating cancers based on these mutations?
That's a major area of progress. Directly fixing or replacing a lost tumor suppressor gene is still really hard therapeutically.
You can't easily give back a function that's gone.
Right. Unlike targeting an overactive oncogene with an inhibitor. But what we can do is target the consequences of the loss.
Like the PRP inhibitors you mentioned for BRCA mutations.
Exactly. That's a perfect example. Cancers with BRCA1 or 2 mutations have a defect in homologous recombination DNA repair. PRP inhibitors exploit that specific weakness. PRRP enzymes are involved in a different repair pathway, and when you inhibit PRP in a cell that also lacks BRCA function, the cell accumulates too much DNA damage and dies. It's called synthetic lethality.
Ah, exploiting a specific vulnerability created by the tumor suppressor loss. Very clever.
It's been a game changer for certain ovarian and breast cancers and now being explored in prostate and pancreatic cancers with BRCA mutations too.
Okay, now let's bring it really close to home for us. Radiation therapy. How does the status of these genes affect how tumors respond to radiation?
It's a critical interaction. Think about P53 again. Its status can really influence radiation response.
How so?
Well, if P53 is functional, radiation damage can trigger it to induce apoptosis or cell cycle arrest, helping kill the tumor cells. But if P53 is lost,
The tumor cells might resist that apoptosis signal?
they might, yes,
Yeah.
making them potentially more resistant to radiation kill through that pathway. However,
It's complex. Oh.
losing P53 can also lead to more genomic instability after radiation. The cells might bypass checkpoints, try to divide with damaged DNA, and then undergo mitotic catastrophe, basically. They die messily during cell division. So loss of P53 isn't always purely radioresistant.
Interesting. What about the DNA repair genes like BRCA or ATM?
Defects there are really important. ATM is another key player activated by DNA double-strand breaks upstream of P53 and BRCA1 in some ways. If tumor cells have defects in genes like ATM or BRCA, they are less able to repair the DNA damage caused by radiation.
Which should make them more sensitive to radiation.
Generally, yes. They accumulate more lethal damage because their repair shops are faulty. This increased sensitivity can potentially improve treatment efficacy.
Is there a downside?
There can be. If the patient has a germline mutation in one of these DNA repair genes like ATM and ataxia-telangiectasia, their normal tissues might also be more sensitive to radiation, increasing the risk of side effects. So it's a balance.
Right. It really drives home that radiation works by causing DNA damage, and the cell's ability to handle that damage, heavily influenced by tumor suppressor genes, is key to the outcome.
Absolutely. It affects how effective our treatment is and potentially informs decisions about dose, fractionation, maybe even the technique we use like IMRT, VMAT, SBRT, SRS, trying to maximize tumor kill while sparing those potentially sensitive normal tissues.
Okay, fantastic overview. This feels like a good moment to maybe crystallize some key takeaways. How about some clinical pearls?
Sounds good. Pearl number one, and it's fundamental. Tumor suppressor genes are the critical breaks.
Yeah.
Their inactivation, that loss of function, is a key, often essential step in cancer development. You really need to grasp that.
Got it. Breaks failing, bad. Pearl 2.
Knudson's two-hit hypothesis. It's the framework explaining how these breaks fail in both sporadic and hereditary cancers. Remember, 2 somatic hits for sporadic versus 1 germline plus 1 somatic hit for hereditary.
2 hits. Crucial concept. And a third pearl.
Loss of heterozygosity, LOH. Remember that this is a super common way to get that second hit in hereditary cancers. It's the mechanism that eliminates the last good copy of the gene. Understanding LOH helps understand how these inherited risks translate into actual tumors.
Excellent pearls. Okay, ready to switch gears slightly. How about we try a quick board blitz to test some of this?
Yeah, absolutely. Let's do it. Fire away.
All right. Question 1. According to Knudson's 2-hit hypothesis, the development of a sporadic retinoblastoma, that's the RB1 gene, requires A. 1 germline mutation and 1 somatic mutation in RB1.
B.
2 independent germline mutations in RB1 is 1 somatic gain-of-function mutation in RB1, or D, 2 independent somatic mutations inactivating both RB1 alleles.
Okay, sporadic means no inherited mutation, and RB1 is a tumor suppressor. It needs loss of function, so you need 2 hits, both somatic. That sounds like D, two independent somatic mutations.
D is correct. Nicely reasoned. Sporadic cases need those two separate somatic events in the same retinal cell line. Okay, question 2. A patient has a known germline mutation in one allele of the APC gene. Which condition are they most likely to develop? A, Li-Fraumeni syndrome. B, hereditary non-polyposis colorectal cancer, HNPCC, also known as Lynch. C, familial retinoblastoma. Or D, familial adenomatous polyposis, FAP.
APC, we link that strongly to the colon and polyps. Li-Fraumeni is P53, HNPCC is mismatch repair, retinoblastoma is RB1. So APC germline mutation points directly to D, familial adenomatous polyposis, FAP.
Correct again. D it is. APC causes FAP. Excellent recall. Question 3. Which tumor suppressor gene product directly regulates the G1 to S cell cycle checkpoint, specifically by binding to E2F transcription factors? A, P53, B, BRCA1, C, PTNN, or D, RB1?
G1S checkpoint binding E2F, that was the gatekeeper function we discussed for. RB1, the retinoblastoma protein, so DRB1.
Spot on. RB1 it is. P53 acts at checkpoints too, but RB's mechanism is specifically through E2F binding. Okay, last one for the blitz. Loss of heterozygosity, LOH, in the context of tumor suppressor genes most often refers to A, the initial germline mutation, B, gene amplification, C, epigenetic silencing of an oncogene, or D, somatic inactivation or loss of the remaining wild-type allele in a heterozygote.
LOH, lost the other copy. So you start heterozygous, one good, one bad, and LOH is the event that gets rid of the good one.
That sounds
exactly. D, somatic inactivation or loss of the remaining wild-type allele.
Perfect. 4 for 4. D is the answer. LOH is that crucial second hit mechanism, losing the normal allele. Nicely done.
Thanks. Those really test the core concepts.
They do. And sometimes mnemonics can help cement these associations. For some of the syndromes we mentioned, maybe think, like father, breast, and colon problems run.
Okay, let's break that down. Li-Fraumeni, TP53, breast, BRCA, and colon. APC, problems run, PTN, and RB.
Yeah, like father, Li-Fraumeni TP53, breast, BRCA, and colon, APC, problems, PTNN, run, RB. It's a bit forced, maybe, but if it helps you link TP53, BRCA, APC, PTN, and RB to their key associations.
Hey, whatever works. Mnemonics can be lifesavers, especially for boards. That's actually pretty clever.
Well, do you think so? Now, before we wrap up, maybe just briefly touch on any ongoing controversies or big advances in this area.
Sure. Well, as we touched on, a major challenge, maybe even a controversy, is how best to therapeutically target tumor suppressor loss. Restoring function directly is tough.
Right, unlike hitting an oncogene.
Exactly. So a lot of effort is on targeting downstream effects or synthetic lethalities, like the PRP inhibitors. But finding ways to actually reactivate or replace lost tumor suppressors, that's still a huge hurdle.
But on the advances side,
definitely the progress in genetic testing. Identifying individuals with germline mutations is becoming much more routine, much more sophisticated. This allows for really personalized risk assessment and management, which is a massive advance in prevention and early detection.
And you mentioned epigenetics earlier. Any advances there?
Yeah, that's a really exciting area. Research into epigenetic therapies, drugs that might, say, remove those silencing methyl marks we talked about is ongoing. The hope is maybe we could reactivate epigenetically silenced tumor suppressor genes in cancer cells. Still early days for many applications, but a lot of potential.
Okay, so let's try to pull this all together then, a quick summary.
Right. So tumor suppressor genes, absolutely essential cellular breaks and guardians. They get inactivated by loss of function mutations, often following Knudsen's two-hit model.
2 hits needed, either both somatic, sporadic, or one germline plus one somatic, hereditary.
Exactly. And LOH is a common mechanism for that second hit. Key players like RB1, TP53, BRCA12, APC, PTN are linked to specific hereditary syndromes when mutated in the germline.
And understanding these genes is crucial for, well, pretty much everything in oncology.
Pretty much, yeah. Cancer risk assessment, genetic counseling, diagnosis, prognosis, and importantly for us, predicting and potentially guiding treatment response, especially for radiation therapy.
Because radiation response is so tied to DNA damage and repair, which these genes heavily regulate.
You got it. It's fundamental RadBio knowledge with direct clinical impact.
Fantastic. This has been a really comprehensive deep dive.
Definitely covered a lot of ground. Hopefully, it helps solidify these crucial concepts.
I'm sure it will. So for everyone listening, remember, you can sharpen your skills further with practice oral boards over at radoncsmartlearn.com.
And definitely subscribe to Rad Onc Smart Review so you don't miss our next deep dive into key radiation oncology topics.
Thanks so much for tuning in.
Yeah, thanks everyone.